Dr. Ecker is a full professor of University of Ottawa, and is in the Department of Biology, and is also a director of the Center of Advanced Research in Environmental Genomics. He graduated at Laval University in 1981 and obtained a PhD title in 1986, okay, at McGill University, one of the top universities in the world. And he is interested in the understanding genetic mechanisms involved in the GABAergic and dopaminergic neuron development, using not only zebrafish as the experimental model, but also a mouse. And however, his pioneering work focused on the helix homologs genes for identifying and characterized their function in the development of GABAergic intelligent populations. So it is the moment to hand over back speaker. Thank you.
Okay, so thank you very much, Julia, for the kind introductions. Great to see you again. I'd like also to thank Paolo and all the colleagues here for the warm welcome. It's a great pleasure to be with you today. So, as Julia said, we do a number of things in the lab. So here, this slide summarizes the various projects we have going on. As Julia mentioned, DLX genes have been for a long time our main object of research in the laboratory, and their role in the development of GABAergic neurons. For a number of years now, we have been also interested in dopaminergic neuron in zebrafish, and that's what I will be telling you about today. But a year or two ago, we started also to use zebrafish to do models of pediatric epilepsy. And as all these projects use a lot of transgenic animals, we also collaborate with colleagues to use the zebrafish to develop novel methods of bioconfinement in for the aquaculture industry. Okay, so these are the various projects, but today, as I mentioned, we're going to look at dopaminergic neuron development, death, and regeneration in the zebrafish. So both the human brain and the zebrafish brain use dopamine as a neurotransmitter. Dopamine is very important as a neurotransmitter. It's important in a number of physiological functions, and one of them is the control of locomotion, and that's why people often associate dopamine neurons with the Parkinson's disease, because the loss of these dopaminergic neurons in humans creates these locomotor dysfunctions. So, in the brain of the human or mammals, we understand fairly well the pathways, the neuronal pathways that are involved in locomotor control. In zebrafish, it is also true that dopamine is involved in the control of locomotion, but the pathways is still a little bit the object of controversy. So in humans, there's a region called the substantia nigra that projects to the caudate and other regions in the basal ganglia as part of this locomotor control. There's no really anatomical equivalent of the substantia nigra in zebrafish, but people for a long time have thought that dopamine neurons in the ventral part of the diencephalon are responsible for these analogous pathways, although there are very good papers that claim that it could be instead neurons that are situated in the telencephalon that do the trick. Okay? But because for a long time we've talked about neurons in the ventral diencephalon, you will see a lot of pictures today that depict these neurons, and you'll see examples of that a little bit later. But why study dopaminergic neurons in the zebrafish? So one good reason could be that, contrarily to mammals, zebrafish, like other fish, have a tremendous regenerative ability. They are able to regenerate several body parts after injury, and that includes the fins that have received a lot of attention, but also includes the heart, the retina, and the central nervous system, spinal cord, and brain. So our central objective, if we were to summarize it, would be to say, to determine if the zebrafish have this ability to regenerate dopaminergic neurons after they've lost them for one reason or another. So the first step in such a process would be to ablate these dopaminergic neurons. And fortunately, there are many ways to do that. Okay? So you can use drugs. And one example would be rotenone, which is a common pesticide that has been associated with an increased probability in humans to develop Parkinson's disease. But there are also the classical experimental drugs such as MPTP, although again in the zebrafish, the effectiveness of drugs like MPTP is questionable. For each of these approaches, I named the students, past or current, who have been doing the work, but I'll introduce these persons as I go along. As you will see, for most of the talk, we are also using a chemogenetic approach using nitroreductase. It's a system that is fairly popular in the zebrafish, but I'll describe it in greater detail in a few minutes. And finally, it is also possible to ablate cells just using a laser, and I'll show you a brief example of that by former graduate student Yan Wei-Shi, although we're not doing this approach routinely anymore. So what I will be telling you about today is 3 lines of transgenic fish that are depicted here, in which we have labeled, I would say, most of the dopaminergic neurons in the brain with fluorescent proteins or with fusion constructs. So I'll show you some examples where we targeted the green fluorescent protein into dopaminergic neurons. So these are the cells that you see over here are these clusters of cells in the ventral diencephalon that, as I mentioned earlier, are thought to be the equivalent of the functional equivalent of the substantia nigra. I will show you also briefly a line of transgenic fish where we targeted MCherry, but not only to the dopaminergic neurons, but to the mitochondria of dopaminergic neurons, because mitochondria have been associated a lot with neurodegeneration associated with Parkinson's disease. And finally, we have a fusion protein between cyan fluorescent protein in blue and the nitroreductase for the chemogenetic ablations, and that will be the last part of the talk. So I'm going to divide the rest of today's presentation according to these 3 lines, starting with this one, and finally the one in the middle. So in order to target proteins to the dopaminergic neurons, we need to use regulatory elements from genes that are specifically expressed in the dopaminergic neurons. And when we started this project, there had been a few attempts to do that using regulatory elements of various genes, such as tyrosine hydroxylase and others. And unfortunately, the success had not been very, very good. So we decided to give it one more shot using the regulatory regions of the dopamine transporter. So here this slide shows you in a very brief way dopamine metabolism. But when dopamine synthesized in the terminals of the dopaminergic neuron is released, it can be reabsorbed in the cell using this dopamine transporter, or DAT, D-A-T, the dopamine transporter. And we decided to use the regulatory elements of this gene, and we were, in fact, not the first ones to do that. Others had attempted that same before. So here is the expression of that, and this horseshoe shape represents these ventral diencephalic dopamine neurons that I mentioned a couple times before. So that's to show you where that is normally expressed. This is in situ hybridization using a DAT messenger RNA as antisense probe. Okay, so Yan Wei-Shi, a former student in the lab, decided to try to target GFP to those neurons, But we had no idea of where these regulatory elements would be. And as I was mentioning, there's one group who had tried that, and the success with that was limited, just trying to use a fragment of the 5' flanking region of that gene. So what Yan Wei decided to do, so instead of trying to identify and characterize these regulatory elements, he decided to just insert the green fluorescent protein in frame with the first exon of the DAT gene. So we took a big fragment of around 30 kilobases, 25 to 30 kilobases of the DAT locus, and we, using homologous recombination in bacteria, we introduced the GFP in frame with the first exon. So we don't know where the regulatory elements are, but we gamble that the fragments that we are using are large enough so that they would be in there. So just to give you an example, the 5' flanking region where regulatory elements are often found is about, the fragment that we used in this particular experiment was about 13 kilobases in length. And we were able to target GFP expression to this horseshoe-shaped structure if we take the ventral diencephalon as an example, and other populations of dopaminergic neurons. And when we do and we compare this with in situ with that, then we see that there is, good correlation between that. And that's what you're looking at here is the expression of the transgene, the GFP, or the dopamine transporter in zebrafish embryos or early larvae. So late embryos, early larvae. But Yan Wei was also able to show that you could detect expression of the GFP in the adult brain. So this is one not transverse, but horizontal section through the brain of an adult zebrafish. And in addition to the olfactory bulb, where there are lots of these dopamine neurons, we see also expression of the GFP in the ventral diencephalon. And it's important because ultimately, as Parkinson's disease is a disease that affects older people, at the beginning we did experiments using embryos and larvae, but we wanted to move into the adult zebrafish as it would be more, arguably, the more representative. Okay, so just to give you an example of the use of these transgenic fish that express GFP and dopaminergic neurons is Yan Wei was able, because you're able to see them, to do specific ablation using a laser. Okay, so here are different planes of view in the brain, and that's done in larvae. And then after ablation, then the cells are gone, and then if you wait a few days, we see that the cells are coming back. So that's showing you one way that these fish can be used, but these experiments are very long and very difficult to do because you need to focus the laser on a large number of cells. And before you are able to see a phenotype, so some effect on the physiology of the fish, you need to ablate a lot of these neurons. Okay? If we make, it's a dangerous analogy, but if we make an analogy with what happens in humans, there is a rough, I'm simplifying, but there's a rough estimate that in Parkinson's disease, you need to have lost more than 80% of your dopaminergic neurons before the phenotype or the symptoms is easily visible. Okay, so we thought, okay, if we want to see something in a fish, we need to have a very good efficiency at ablating these cells. Okay, so that was an example of these that GFP fish. Just to give you the fish became popular, so that was the one good thing about them. We've distributed these fish to more than 50 labs so far. So that, so I think people like them. Okay, so Parkinson's disease is associated often with altered mitochondrial function. So we thought it would be kind of interesting to see if we could label the mitochondria specifically in dopaminergic neurons. So that's what we did. Use the same strategy as to make that EGFP fish, except that here, instead of EGFP, we used MCherry, but a fusion protein with the mitochondria localization signal of a gene called TOM20. So this would target, MCherry to the mitochondria, and the regulatory elements of the DAT gene would target this entire thing into the dopaminergic neurons with patterns that are closely similar to the ones we had obtained with EGFP. And that's work that Sandra Noble, a former PhD student in the lab, did. And here on the right, you see expression of MCherry in those fish, either dorsal or lateral views. And then if we do cell fractionation experiments to see where MCherry is located, we observe that MCherry is mainly made in the mitochondrial fraction of the cell as opposed to the cytoplasmic fraction of the cell. So that was very good, but then the purpose at the beginning was to be able to observe the mitochondria in dopaminergic neurons. And then if you affect the life of these dopaminergic neurons, what happens to the mitochondria? So mitochondria, I'm going to simplify, but are dynamic structures. They have the possibility of fusioning to make fusion mitochondria, but also, depending on the physiological conditions, they can separate, so there can be fission of the mitochondria. And Zong Nok, a current graduate student in the lab, has decided to look at that. Now, mitochondria are small, so the imaging of the mitochondria in these fish is difficult. In order to be able to see if the mitochondria fuse or fission, it was a very challenging project for Zong. So here's a normal fish, and this is imaging of the mitochondria. It's green, but it's MCherry you're looking at using an antibody. And then what Zong did is he treated the fish with rotenone, which is this pesticide that is associated with Parkinson's disease, and that is known to alter complex 1 function in the mitochondria and cause the death of dopaminergic neurons. And Zong looked at the morphology of the mitochondria, and as the arrowheads point out, he saw a large increase in the frequency of mitochondria that had undergone fission, so that had separated. Okay?